So what if life isn't? Well, isn't always like we know it. Thriving on water and oxygen. Imagine these worlds where water is basically rock solid and the air is just this thick orange haze. Could life actually exist there? Like really challenging everything we assume about what makes a planet habitable.
Yeah, that's a really profound question, and it's precisely what we're going to embark on today. Welcome, everyone, to a deep dive into this, well, fascinating world of alternative biochemistries and extreme environments.
Right, our journey today really starts by challenging everything we thought we knew about life's possibilities. We're exploring sources that really push the boundaries of what makes a planet habitable. Think methane-rich moons, the deepest parts of Earth.
Hmm.
All to see if life might be hiding, maybe even in plain sight, right here in our solar system, especially, on Mars.
Mm-hmm. And we've pulled together some cutting-edge astrobiology research, scientific papers on Earth's own extremophiles, and new analyses looking at potential Martian life.
Yeah, the goal is to give you a shortcut, basically, to being really well-informed about where astrobiology is heading.
Okay, so to really stretch our imaginations, let's turn our attention first to Saturn's largest moon, Titan. It's a world that, at first glance, seems utterly hostile to life as we know it, but it holds some really tantalizing clues.
Titan's just fascinating, isn't it? It's got this nitrogen-rich atmosphere, thick with hydrocarbon smog, almost soupy. But what really grabs you are those images from Cassini and the Huygens probe. They show a landscape with clear signs of, well, running liquid. Drainage channels, lakes even. It almost looks Earth-like in certain ways.
It is Earth-like in that specific sense, yeah, with a liquid cycle.
Gotcha.
But the crucial difference, and it's a big one, is the temperature. Titan dips down to a frigid 94 Kelvin. That's about minus 179 degrees Celsius. Super cold.
A minus 179 C. Wow.
Yeah, at those temperatures, water is literally rock hard. So the liquid flowing and pooling on Titan's surface, it isn't water at all. It's liquid methane and ethane.
Liquid methane.
And that single fact just completely redefines our baseline for a habitable world. It challenges that whole notion that water has to be the universal solvent for life.
So, okay, if water is rock, what's this liquid methane doing? And how does that open up possibilities for a totally different kind of life cycle?
Well, just like water on Earth, methane on Titan has what we call a triple point. That means it can exist as a liquid, a solid, or a gas in its natural environment. And this allows for a full methane cycle. You get methane clouds, methane rain, methane lakes, and rivers.
A whole weather system based on methane.
Exactly. And Chris McKay, a researcher at NASA Ames, he's speculated on what potential methane-based life might look like there. He suggests that hypothetical local methanogens, methane-based life forms, could maybe consume hydrogen, acetylene, and ethane from the atmosphere, and then exhale methane gas.
Okay, and the interesting part is that this would leave a specific chemical signature, right?
Precisely. A depletion of those consumed gases, hydrogen, acetylene, ethane, right there on the surface, you'd expect less of them if something was eating them.
And Titan actually shows some hints of that depletion. I remember reading about that.
It does show hints, yeah. But it's definitely not conclusive proof of life. Not yet, anyway. Our atmospheric models for Titan are still evolving, and there could be other non-biological processes that mimic these characteristics.
Right, other explanations.
Yeah. As Lisa Kaltenegger from the Max Planck Institute put it, we just don't know what the telltale signs for life would be in such an atmosphere because it is so vastly different from ours.
So the jury's still out, which makes you really want to send more probes there, doesn't it?
Absolutely. Future probes are incredibly desirable. Imagine surface rovers, or maybe aerostats like balloons floating in the atmosphere, getting a really focused look, sniffing out these specific depletions, perhaps.
Okay, so if methane could potentially support life right here on Titan, what does that imply for planets outside our solar system? Does this massively expand the hunt for life?
Oh, it absolutely does. If you add methane into the mix as a potential life-supporting liquid, it dramatically extends the traditional habitable zone around stars. Planets much further out from their star, where water would be frozen solid, could potentially host methane-based life. This just vastly increases the number of potential candidates.
So we should be looking further out.
Exactly. And M-dwarfs, those red dwarf stars, they become really prime targets for study. Their planets orbit closer in, which makes them easier to detect with our current methods.
Makes sense.
Yeah, take a star like Gliese 581, for example. A potential methane habitable zone around it could exist somewhere between, say, 99 and 248 million kilometers out. Now, Gliese 581 does have confirmed planets, but none are quite in that specific methane sweet spot.
Not quite, right?
Not quite. For a smaller M4 type dwarf star, that zone is even closer in maybe 12.6 to 34.4 million kilometers.
Then there's something about the light from red dwarfs too, isn't there? More infrared.
That's right. Red dwarfs emit more red and infrared light, and that type of light could actually warm a Titan-like planet more effectively than our sun's light warms Titan, potentially extending that liquid methane zone even further.
Huh. And here's a really wild thought from the sources our own sun.
Yeah.
When it becomes a red giant like 5 billion years from now, it might briefly warm Titan enough for a temporary methane habitable zone.
Whoa. A fleeting window of opportunity before the sun expands further. That is a wild thought.
Strange, right? So let's speculate a bit more. What would this methane life actually be like? The temperatures are incredibly cold.
Right. The frigid temperatures on Titan, that minus 179 C, would strongly suggest incredibly slow metabolisms, life operating on timescales perhaps almost unimaginable to us. Chemical reactions just happen much, much slower when it's that cold.
You mentioned the slow metabolism. But what about methane itself as the liquid, the solvent? How does it compare to water? Are there unique challenges or maybe even advantages?
That's a really crucial point. There's a big debate here. Water has this unique property. It expands when it freezes.
Right, so ice floats.
Exactly. Ice floats, lakes freeze from the top down, insulating the liquid water below, which is vital for aquatic life on Earth during winter. Methane doesn't do that. Methane is actually less dense as a liquid than as a solid. So methane ice would sink.
Okay, so that sounds like a problem. A lake would freeze solid from the bottom up.
It might seem like a drawback, yeah. But there's a potential flip side.
Oh.
Methane not expanding when it freezes could mean that cells wouldn't necessarily rupture if they froze solid. Life might be able to go dormant when frozen and then revive upon thawing without the cell damage water ice causes.
Ah, so it's just a different set of rules, a different survival strategy. Fascinating.
Indeed. It forces a fundamental shift in our assumptions. You know, science fiction writers like Isaac Asimov even speculated about this stuff, life in methane based on complex lipid molecules forming membranes in cells. And some researchers argue that free radical reactions, which are possible at these incredibly low temperatures, might allow methane-based life to be only fractionally more sluggish than Earth life, not astronomically slower.
What about energy, though? That seems like a big hurdle. Methane-based chemistry probably has a much lower energy yield than the oxygen-based chemistry that powers complex life here.
That's a valid concern. The energy flux is likely lower, but there could be counterarguments. For instance, hydrogen, which is part of that hypothetical methane life cycle we talked about. The stuff they might consume actually diffuses much faster than oxygen does.
Okay.
So that faster diffusion could potentially make up for some of that lower energy yield by allowing for more rapid transport of the reactants needed for metabolism.
So speed of delivery might compensate for lower energy per reaction.
Possibly. We can even sketch out a hypothetical methane-based biochemistry. Respiration could involve inhaling hydrogen, maybe reacting it with something like acetylene, and exhaling methane, perhaps water too. And photosynthesis could work in reverse splitting methane and water using sunlight, releasing hydrogen gas.
Wow. It's truly an alien vision of life, but grounded in, possible chemistry and physics.
Exactly, which brings us back closer to home, actually, back to Earth.
Right, because Earth itself hosts life in conditions that were once thought completely impossible, these extremophiles.
Precisely. They completely reshape our understanding of what habitable even means. They prove that life is far, far more adaptable than we ever imagined.
I think studying them is just critical for astrobiology, isn't it?
Absolutely critical. They give us profound insights into how life might have originated here, and crucially, what the potential for life is beyond Earth. There are real-world test cases for the limits of biology.
It really is mind-boggling. It's like these organisms didn't get the memo about the rules. They just find a way. If you had to pick, which adaptation just blows your mind the most?
That's a tough one. They're all pretty incredible. But okay, let's take a quick tour and you'll see why it's hard to choose. First up, you've got thermophiles and hyperthermophiles. Heat lovers.
Found in hot springs, deep sea vents, that kind of thing.
Exactly. Places like Yellowstone or those black smokers miles down in the ocean. Their cells have amazing adaptations. Changes in cell walls, changes in the fats, the lipids in their membranes, making them more saturated, more stable at high heat. And special proteins, often helped by chaperone molecules that resist unfolding or refold correctly if they do get too hot.
And their spores. I read they're considered maybe the toughest known form of life.
Spores are incredible, yeah. Bacterial spores especially. They can survive unbelievable heat, radiation, chemicals. Their resistance comes from having very little water inside the core, special protective proteins coating their DNA, and unique chemicals, like tiny armored vaults for genetic material.
Amazing. Okay, so that's heat. What about the other extreme? Cold.
Right. Then we have the psychrophiles, the cold lovers. These are microorganisms that thrive at low temperatures, often optimal growth around 10 degrees C or even below freezing. We find them in glaciers, deep oceans, polar regions, and incredibly viable cells have been found in glacial ice cores after being frozen for over 120,000 years.
120,000 years. Right. Frozen solid. That just changes everything about looking for life on icy moons or planets.
It really does. It shows life can essentially pause for geological timescales and then potentially revive. They manage this by keeping their membranes fluid with special lipids and reducing the viscosity inside their cells.
And didn't you mention some Antarctic bacteria with built-in flotation devices?
Yes. The gas-vacuolated sea ice bacteria, they have these tiny gas vesicles inside them which act like little life jackets, helping them float up or down in the water channels within the sea ice to find the best light conditions. Ingenious.
Okay, heat, cold. What else? Acid.
Yep, acidophiles. Loving low pH. They thrive in highly acidic conditions, optimally below pH 3. Think acid mine drainage, volcanic springs. They often have protective outer capsules or layers, very impermeable membranes to keep acid out, and active systems to pump protons out, maintaining a more neutral pH inside the cell, creating their own little safe space.
Then there's pressure, right? Deep ocean trenches.
Exactly. Barophiles or piezophiles. Pressure lovers. These tiny things live way down, like in the Mariana Trench, nearly 11,000 meters deep. The pressure there exceeds 1,000 times atmospheric pressure, crushing.
How do they even survive that?
Their cell membranes are adapted. They often have a higher proportion of unsaturated fatty acids, which keeps the membranes fluid and permeable under that immense pressure, preventing them from becoming rigid or collapsing.
Incredible. What about dryness? Lack of water?
That brings us to xerophiles and xerotolerant organisms, dryness survivors. Found in deserts, salt flats, places with very little available water. Their strategies include things like shrinking their cells to minimize water loss and accumulating special protective molecules like LEA proteins, late embryogenesis, abundant proteins which help prevent cellular damage and protect DNA from desiccation stress. Basically, they go into a kind of suspended animation until water returns.
And finally, salt lovers.
Halophiles. They thrive in high salt environments like the Dead Sea or salt evaporation ponds. Too much salt draws water out of normal cells, but halophiles have ways to cope. They accumulate high concentrations of compatible solutes, things like potassium ions, sugars, or amino acids inside their cells. These osmolytes balance the external saltiness, allowing the cell to retain water.
And one of these, halobacterium you mentioned, has modern applications. Something about light.
That's right. Halobacterium produces bacteriodopsin. It's a protein that captures light energy and uses it to pump protons, generating energy for the cell. It's a kind of simple photosynthesis, and this pigment is remarkably stable and has unique properties. NASA has actually explored using films made of bacteriodopsin to help illuminate gas spill locations in space. Old microbial trick. New space-age application.
It shows how studying these extreme organisms isn't just academic.
Not at all. It goes way beyond theory. Extremophiles are hugely valuable. Thermostable enzymes from thermophiles, like Taq polymerase used in PCR for DNA amplification, are cornerstones of biotechnology. That's a multi-billion-dollar market right there, used everywhere from medical diagnostics to crime scene investigations. Plus, we're finding potential new antibiotics, industrial catalysts. They're a treasure trove for humanity.
Okay, so armed with this knowledge of Earth's extremophiles, let's bring it back to Mars, specifically the idea of life beneath the surface.
Right. The whole Martian methane mystery, that methane appearing and disappearing in the atmosphere, has kept speculation alive for years. Is Mars geologically active or maybe biologically active?
And while there are non-biological explanations being proposed now, like electrical discharges in dust storms generating methane over ice,
the possibility of life, particularly microbial life, remains intriguing. And that brings us back to methanogens, those methane-producing microbes. Could Earth-like methanogens survive on Mars?
especially the hydrogenotrophs, right? The ones that eat hydrogen and carbon dioxide.
Exactly. Those are prime candidates. They feed on simple inorganic molecules, hydrogen gas, H2, and carbon dioxide, CO2. Both are expected to be available, at least potentially, in the Martian subsurface. And crucially, those ingredients can have an abiotic origin, meaning that they can be produced by geology, not just by life. That's important for getting life started in the first place.
So people have actually tested this. Put methanogens in simulated Mars conditions.
Oh yes, extensive lab studies have been done. Researchers basically create Mars in a jar conditions, mimicking the low temperatures, the dryness, the salts, the atmospheric pressure, even the radiation, and see if terrestrial methanogens can survive or even grow. These experiments help us map out the absolute limits for Earth life under Martian conditions. It gives us a kind of roadmap for where life might be possible.
So to find potential Martian habitats, we look for similar places on Earth, analogs.
Precisely. We study Earth's own extreme subsurface environments. The sources highlight three main types as good Martian analogs. One, deep fractures in continental bedrock, like deep mines or boreholes. Two, subglacial lakes and brines, like Lake Vostok in Antarctica. And three, deep sea hypersaline anoxic basins, super salty, oxygen-free basins on the seafloor.
Places where life is really pushed to its limits here. What have we learned from those analogs?
Well, studies show methanogens are found in many of these sites, about 2/3 of them, according to the data compiled, but usually in pretty low abundance. There was one site, Liddy Hot Springs, that showed an ecosystem almost entirely dominated by a single type of methanogen, which is interesting, but that seems to be the exception.
So normally they live in communities.
Yeah, typically they coexist in complex microbial communities. Different types of microbes living together, often depending on each other for nutrients or waste removal. It's rarely a one-bug show.
And the specific conditions determine which types of methanogens thrive.
Absolutely. Temperature is a big one. We find methanogens tolerating an incredible range on Earth, from below freezing, around -17.5 C, all the way up to 82 C in hot springs. In the colder analog sites, families like Methanosarcinaceae and Methanomicrobiaceae tend to be more common. Others prefer warmer conditions.
What about salt? Mars is thought to have lots of salts.
Salinity is key. Some methanogens, particularly certain hydrogenotrophs, prefer lower salt levels. But others, especially within that Methanosarcinaceae family, are incredibly salt tolerant. They can handle brines up to 9 molar ionic strength that's incredibly salty, way saltier than seawater. This is really relevant for Mars because salts can lower the freezing point of water, potentially keeping pockets of liquid brine available even below 0 degrees C.
Makes sense. And pH does that vary too.
It does. Methanogens tolerate a pretty wide pH range overall, maybe 4.5 up to 11.3. But again, different groups have preferences. Methanosarcinaceae and Methanomicrobiaceae are often found in more neutral to acidic waters, while some others like more alkaline conditions.
Okay, one more factor mentioned was sulfate. Mars is sulfur-rich. How does that play in?
Right. Sulfate is important because sulfate-reducing bacteria can compete with methanogens for the same food source, hydrogen. Some methanogen families are inhibited even by low sulfate levels. But again, Methanosarcinaceae seem to be champs here, tolerating significantly higher sulfate concentrations. Their tolerance to both high salt and sulfate really makes them stand out as candidates for Mars.
So putting all this together, where on Mars might we find a potential sweet spot for these kinds of microbes? We know there's ice near the surface, maybe even near the equator now.
Well, first you need energy. Hydrogen is key. Some might diffuse down from the atmosphere where it's made by sunlight breaking down water vapor. But a potentially more significant source deep down is radiolysis.
Radiolysis?
Yeah, the splitting of water molecules by natural radioactivity. Rocks contain radioactive elements, thorium, uranium, potassium. Mars has these in its crust at levels similar to Earth's deep ocean basalts. This radioactivity can break apart water molecules trapped in the rock, releasing hydrogen gas, a steady long-term energy supply, independent of sunlight or surface conditions.
A deep underground energy source.
Exactly. Other processes like rock friction during fault movements or water rock reactions like serpentinization can produce hydrogen, but the geological evidence for widespread recent serpentinization on Mars's surface is limited, maybe except for specific areas like Nili Fossa. Radiolysis might be more widespread underground.
Okay, so based on energy potential and likely water, any specific regions look good?
The research points towards the southern Acidalia planitia as a particularly promising target region for future missions.
Why there?
Because orbital data suggests it has a high abundance of those heat-producing radioactive elements, and it's likely to have significant subsurface water ice. Combine heat production with water ice, and you increase the chances of finding liquid water deeper down.
And how deep are we talking?
The models estimate that the 0 degrees C isotherm, the depth where the temperature hits freezing point, allowing ice to potentially melt, could be found somewhere between 4.3 kilometers and 8.8 kilometers deep in Acidalia planitia. The exact depth depends on assumptions about how deep the ice or water goes.
That's deep. Kilometers down.
It is deep. So the best candidates for life in those potential cold, deep Martian environments would likely be cold-adapted methanogens, similar to Earth's Methanosarcinaceae and Methanomicrobiaceae. And importantly, they'd probably exist in those centrophic clusters we talked about, communities of different microbes working together rather than just single species dominating. Life, even in extreme places, often relies on teamwork.
But it's not a slam dunk. There's still big challenges and unknowns, right?
Oh, absolutely. Huge gaps in our knowledge remain. We don't really know how much inorganic carbon, like CO2, is actually available deep down in the Martian subsurface. We need much better data on the actual depth of water or ice, the porosity of the rock, how fractured it is, all things that affect whether liquid water can exist and microbes can move around and get nutrients.
And the competition factor.
Right. Competition for hydrogen, especially from sulfate reducers, could be significant. Depending on the exact conditions, sulfate reduction might be energetically more favorable than methanogenesis. And then there's the perchlorate problem.
Ah, yes. Those salts found all over Mars.
Exactly. Perchlorates are highly oxidizing compounds. They make the environment much harsher for life as we know it. Studies show they severely reduce habitability for terrestrial microbes, including methanogens, even at relatively low concentrations above about 0.03 molar ionic strength. So any potential Martian life would need strategies to either avoid hyperchlorate zones or somehow detoxify these compounds. That's a major hurdle.
Wow. Okay, so we've really journeyed from the truly alien possibilities on Titan to the limits of life on Earth, and then applied that to the deep, hidden potential of Mars. You've just taken a deep dive into the incredible possibilities of life beyond our familiar water-based biology.
We really have. We've seen how Earth's own extremophiles just shatter our old definitions of habitable, pushing the known boundaries of survival.
And how those insights are absolutely crucial for guiding our search for life elsewhere, whether it's on icy moons like Titan or deep beneath the red sands of Mars.
So, maybe a final thought to leave you with. If life can find a way in such wildly different and challenging conditions right here on Earth, from superheated volcanic vents to deep, freezing, salty brines, what does that truly suggest about how common life might be out there in the vast, diverse universe?
Yeah. What stands out to you, listening now, as the most surprising adaptation we discussed? And how might thinking about this, about methane lakes or deep Martian microbes, change your view of our own cosmic neighborhood?